Hi again. So now we're going to continue our discussion of functional association networks, and I'm going to demonstrate to you a tool that we developed in the lab called Genes2FANs. So before we came up with the sets to networks algorithm, we were able to create functional association networks from gene set libraries using two other methods for assessing the similarity between pairs of genes based on the co-occurrence in gene sets or in gene set libraries. And for that we created the tool Genes2FANs that can help you to build subnetworks that connect your own gene sets with connections that were found through functional associations based on the processing of gene set libraries. So for Genes2FANs, what we've done, we took 13 different gene set libraries, the connectivity map from CMAP, which I will discuss later and explain to you more about what is that. The CHIA database, which we're also going to discuss more in detail, which is a chip enrichment analysis tool or a gene set library created from publications that reported ChIP-seq experiments. Gene riffs, which I'm going to describe to you in a couple of slides in this lecture. Then we also created a gene set library from the gene ontology. We also created a gene set library from TransFac. We also processed the MGI mouse phenotype browser and created a gene set library from that ontology. And we also created a gene set library for proteins that share structural domains from PFAM and Interpro, as well as genes that are associated with various diseases from OMIM, and then drug targets for FDA-approved drugs. So those gene set libraries were processed. First, we created the gene-gene association computation. We used two other methods that are not as good as the sets to networks method, but they perform relatively well. One is the Jacquard index, and the other one is the binomial proportion test, which I'm going to describe to you. We applied some arbitrary cutoff before we showed how with sets to networks we are capable to find where we should probably put a cutoff. Here we just made an arbitrary cutoff for those scores that compute the association, the potential association between the pairs of genes from those gene set libraries. And then we also have another step, which is a declustering algorithm that tries to deal with the fact that those functional association networks, once created using those methods, tend to have very dense clusters, and the highly connected nodes are also connected. But I'm not going to go into the details of how this declustering algorithm works. And then we have those final functional association networks for each of those categories. So just to go over the Jacquard index, the Jacquard index can be used to assess the similarity between pairs of genes across an entire gene set library. The Jacquard index is a measure of the similarity between two sets. You normalize the overlap between the two sets, so you compute the intersection and divide it by the union. This will give you a number between 0 and 1. You can also sometimes subtract that number from 1. One of the disadvantages of the Jacquard index is that it does not work very well when you compare two sets of various lengths. So for example, if you have one set that has only 5 genes and another set of, like, let's say 100 genes, you can have 100% overlap between the 5 and the 100. So those 5 genes will be in the 100, but still you can get a relatively low Jacquard index score. For that, there are methods for set similarity that can be used that compute probability, and those are more accurate when the lists are of various size. To assess the similarity between genes, we transposed the gene set library, where now each gene is the set label and then the terms are the entries, and this way we can now compare every pair of genes based on the functional terms that the genes share. So I mentioned that it's probably when the set size are not equal, it's better to compute probability. And one way to approximate the overlap between two sets is to compute the binomial proportion test. And this test is an approximation of the Fisher exact test, which we're going to discuss in details later on. And what it does, if the proportions are large enough, it computes the z-score, or the probability of the two sets to overlap at a certain level. So you have the two sets size, a contingency table problem. So a contingency table usually has 4 entries. One would be the overlap between the two sets. One would be the set size of each set, the maximum number of possible overlap that can be between the two sets. And then the 4th entry in the contingency table would be all genes, or all possible genes that can be identified within those sets. And then you just plug in the numbers and compute a probability that is an approximation of the real binomial probability to have the overlap that you observe in this particular case. And this was also used to compute functional similarity between genes within each of those gene set libraries. This is the result from the study. On the right you can see the Genes2FAN website, which we're going to go to now and try to show you how it works. And we have the visualization of the various functional association networks that were created with Genes2FANs. One of the other networks that we added to this application is a protein-protein interaction network, where we consolidated interactions from various databases to create a background protein-protein interaction network. Once you have those functional association networks, or those protein-protein interaction networks, you can use those for various types of analysis. You can look at the structure of those networks, and in some of those networks you can really see that there are various clusters that could be potentially interesting from a functional perspective of understanding the global topology of mammalian cells. But what we are really interested in those networks for at least for the time being, is to query gene lists. For example, if we identify differentially expressed genes, or if we identify genes from other types of proteomics or genomics experiments, we can try to identify if they have some functional connection as a group. And we're going to spend a lot of time in this course trying to analyze those type of genes, and this is just the first example of a tool that tries to do it. So now let's go online and look at how to use the Genes2FANs tool. So in Google you can just type in Genes2FANs, and the first entry should direct you to the website where the application is hosted. And this is the Genes2FAN application. So one of the other features of Genes2FANs is it has a PubMed query that can help you to identify gene lists automatically from PubMed instead of providing your own gene lists. So if you have your own gene list, you can go to this See Genes entry and click on Add List, and that pops up a window where you can cut and paste your genes. However, since we don't have a gene list for this demo, we're going to use the Query PubMed feature of Genes2FANs to identify a gene list. And right now it sets to limit to 20 genes. So what we're going to do, we're going to type in a general term, and that general term will query PubMed to identify publications that mention the term breast cancer. And then using GeneRiff, which I'm going to explain to you in a second, the tool identifies the top 20 genes that are mostly mentioned in publications that were returned with the query breast cancer. So a few words about GeneRiff. So this GeneRiff stands for Gene Reference into Function. It's a manual effort by NCBI curators to associate papers with genes and with some function. So it's a text file that you can download from the NCBI FTP website, and there is some more description about what it is in this particular URL. Here is a download, the header of the file, downloaded and opened in Excel. As you can see, you have the tax ID, which identifies the organism, the gene ID, which identifies the gene for that organism, as well as the PubMed ID that is associated with the gene. So in Genes2FAN, what we've done, we have the query, we query PubMed, we get all the PubMed IDs, and then we count the number of times genes appear in those PubMed IDs that were returned with the general search terms that you entered in that text box, where I just showed you an example using breast cancer. So we have a network that the tool created, potentially interact with it. We can zoom in and out. We can move the locations of nodes. And this is our breast cancer functional association network that was created using the Query PubMed feature. You can see that right now only the protein-protein interaction fan is selected. So all the links in this subnetwork are protein-protein interactions. However, we can change this and select some other types of functional association links, and each one of them has its own color. And then we can run again, and now we are going to see a network that is connected based on those functional association links and not based on the protein-protein interaction links. So the program identified a complex that is made of ABC subunits, and those are connected through a shared biological process. So the ABC are ATP binding proteins that are part of a membrane transporter, and here the connections between those genes and associate other members that are not necessarily associated with breast cancer came up into forming this complex. The Genes2FANs tool uses the Flash network viewer that we developed for visualizing small networks. However, you have other options to view the networks that are generated by this tool, and those are listed here. One is an XML format, and another one is a GraphML format, which I'm going to download. One more feature of Genes2FANs that I would like to mention is that it uses the shortest path algorithm to find connections between the genes using those background functional association networks. So in the options on the left, you can choose the path length, the shortest path length. And if you have a short list of genes and you don't get a lot of connections, you probably want to change that to a higher path length. However, if you have a long list of genes, you probably want to lower it to path length of 1, and this will just find connections between your input gene list. The tool also computes the significance of those additional genes or proteins that get pulled in to the functional association network. It computes the probability of having those additional components entering the network. And this is also computed with the binomial proportion test based on the number of links the node has in the subnetwork that we just created, as well as the number of links the node has in the background functional association networks. In general, I do not recommend using Genes2FANs for your analysis. We are going to show you more advanced methods to analyze gene lists, as well as to visualize networks from gene lists.